Welcome to SciFlux. Here you get daily coverage of the latest scientific research. Every day we bring you new research in a clear and simple format, so you can stay informed without wasting time. Like the video, subscribe, and leave a comment to stay connected and help this channel grow. And if you want each new research delivered directly to you, join the SciFlux newsletter. Let's start by sharing that researchers explored how an active bath of enzymes can influence liquid-liquid phase separation, LLPS, the process where certain proteins form droplets inside cells. Using urease, an enzyme that speeds up in the presence of its substrate, they tested its effect on ubiquinol-2, a protein that naturally condenses under higher temperature or salt conditions. A microfluidic device created a controlled environment, maintaining steady reactions while removing byproducts. This setup isolated the physical impact of enzyme activity from chemical effects. The results were striking. The active enzyme bath produced protein droplets that were larger, denser, and more concentrated. Simulations showed this occurs because enzyme activity acts like an effective temperature, boosting molecular motion and driving phase separation. This study is the first to demonstrate that enzymatic activity alone can physically modulate LLPS, offering new insight into how active enzymes might shape the inner organization of living cells. Next up, researchers tackled the challenge of observing molecules in motion, from drug binding to protein folding to material deformation, using molecular dynamics. While this method shows every atom in action, it's far too slow for large or complex systems. To speed things up, coarse-grained models group atoms into simpler beads, but this simplification introduces noise, making AI-trained force predictions less accurate. To solve this, the team applied knowledge distillation. A teacher, neural network, first learns the physics from noisy data. Then a student network learns from the teacher's refined output. Using multiple teachers further improved accuracy. Tests on a complex liquid system showed this approach produced stable, precise force fields that closely mirror real molecular behavior. By teaching AI to denoise molecular simulations, scientists can now model larger systems over longer timescales, accelerating drug design, materials development, and our understanding of molecular processes. Moving forward, researchers are exploring a world where broken bones and failing organs can be repaired or replaced seamlessly, thanks to biomaterials, specially engineered substances that interact safely with living tissues. For decades, scientists have sought ways to make implants compatible with the body, and that vision is now coming to life. Biomaterials take many forms: metals, polymers, ceramics, and composites, tailored for strength in bones and joints or softness in blood vessels and organs. They already transform medicine. Pacemakers regulate hearts, stents open arteries, and dental or joint implants restore function. They also enable tissue engineering, advanced drug delivery, biosensors, and even improved imaging in CT and MRI scans. Their true power lies in adaptability. By matching biological and mechanical needs, biomaterials extend life and enhance health. At the intersection of engineering and biology, they're shaping a future where healing and technology merge, redefining what's possible in medicine. Next up, researchers examined whether it's possible to build a perfectly lean production system from scratch, a factory with no legacy issues and maximum efficiency from day one. They studied a Brazilian metalworking company setting up a new manufacturing line designed to be lean from the start. The findings were clear: even brand new systems aren't lean immediately. Lean performance emerges over time, progressing through stages from initial design to ramp-up and finally steady operation. Adaptability, not instant perfection, drives success. By applying the theory of constraints, identifying bottlenecks and managing buffers, the researchers developed a framework showing how lean principles gradually take hold. They also emphasized that new systems shouldn't be treated like routine improvement projects. Uncertainty is inherent, and strategies like design to adapt and prepare and adapt allow the system to evolve effectively. The key insight: operational excellence grows through learning, adaptation, and refinement. True lean systems aren't simply designed, they are evolved. Moving forward, researchers are exploring materials that do more than replace tissue. They actively promote healing. Bioactive glasses are at the forefront of this effort, offering a revolutionary approach to regenerative medicine. Unlike traditional implants, these glasses interact with the body to stimulate bone growth and tissue repair. A recent review examined three main types: silicate, borate, and phosphate-based, highlighting how each contributes differently. Silicate glasses bond strongly with bone and guide growth, making them ideal for grafts and implants. Borate glasses offer antibacterial properties and enhanced cell proliferation, supporting soft tissue healing and infection prevention. Phosphate glasses closely mimic natural bone minerals, boosting cellular-level regeneration and bone repair. This research underscores that selecting the right type of bioactive glass is critical for treatment success. As these materials continue to evolve, the vision of implants that restore function and regenerate tissue from within is becoming a tangible reality. Next up, researchers are merging nature, nanotechnology, and medicine to create powerful, sustainable solutions. By harnessing the medicinal plant Vicoa indica, they've developed zinc oxide nanoparticles, ZNONPs, without using harsh chemicals, making the process eco-friendly and efficient. The method is straightforward. The plant's leaf extract reacts with a zinc solution, naturally forming nanoparticles. Characterization confirmed their spherical shape and pure composition. Testing revealed impressive biological activity, strong antibacterial effects against Staphylococcus aureus, potent antioxidant properties, and remarkable anti-cancer potential, reducing lung cancer cell growth by over 85%. This study highlights how plant-based nanotechnology can produce advanced, life-saving materials, showing that sustainable approaches can drive the next generation of medical innovation. Moving forward, researchers are combining drug delivery with tissue regeneration through advanced bioactive nanofibers. They engineered ultra-thin fibers from biocompatible polymers, PCl and PVP, loaded with Amoxicillin, and enhanced them with bioactive glass, including a surface-modified version for optimal performance. Testing revealed that the enhanced fibers absorbed water better, strengthened structurally, and promoted hydroxyapatite formation, critical for bone repair. The modified fibers achieved nearly 96% drug loading and released antibiotics in 2 phases, an initial burst to combat infection, followed by sustained delivery over 2 months. These multifunctional nanofibers represent a leap in wound care and bone healing, acting as smart dressings or implant coatings that actively fight infection while guiding tissue regeneration. It's a clear demonstration of nanoscale engineering, turning materials into active partners in healing. Next up, researchers tackled a pressing question about 5G networks: how to reduce human exposure to radio waves, measured by specific absorption rate, SAR. They designed 5G antenna operating at 9.1 GHz and paired it with a tiny 18mm energy absorber, 2 copper rings on a circuit board. Using simulations near a virtual human head, they tested how effectively the absorber could block or dissipate energy. The results were striking. The device created multiple stop bands, sharply reducing energy reaching sensitive areas like the ear and brain, and significantly lowering SAR. This innovation points towards safer 5G technology, showing that faster connections don't have to come at the cost of exposure, bringing us closer to a future where speed and safety coexist. Finally, researchers explored a safer way to keep food fresh longer by tackling microbial spoilage, a leading cause of waste and illness. They turned to silver nanoparticles for their antimicrobial power, but avoided traditional toxic forms by using pumpkin seed coats, a natural discarded material, to create biogenic silver nanoparticles. These were incorporated into a polymer film to form a new type of smart food packaging. Tests showed the film was thermally stable and effectively inhibited harmful bacteria like Listeria and Salmonella, as well as spoilage fungi such as Aspergillus and Penicillium. Early toxicity tests indicated some effects on small organisms, highlighting the need for further safety studies. Even so, this research demonstrates a promising step towards sustainable antimicrobial packaging, a potential solution that extends shelf life, reduces waste, and protects the environment. Thanks for watching. If you enjoyed the video, please like, comment, and subscribe. Also, join our newsletter to stay informed with the latest discoveries shaping science and innovation. See you tomorrow with a new round of breakthroughs. Take care.